Hello dear learners, in this video we are going to understand about the colony hybridization method, and that colony hybridization method we need to understand, although with the help of a diagram, but we will also cover the theory together so that we can understand that what does this method mean and what is it used for. So basically in the colony hybridization method, what you're trying to do is that you're trying to grow multiple bacterial colonies on a plate, and this plate is called as a master plate. Now this bacterial colonies will grow from a single bacteria which is transformed. Now what does it mean? So I'll explain you here. Let me take this side. So you will take the bacterial cells. Now let's label them from different colors. Suppose one is red, one is green, the another one being purple and whatnot. So what you are going to do is that you are going to introduce your gene of interest, GOI, into this also, into this also, into this also. Now this gene of interest, when it is introduced into the bacterial cell, the process is known as transformation. So in the transformation, you are actually trying to introduce your gene of interest into the bacterial colonies or bacterial cells. And this can be done by using certain vectors which will help you to introduce your gene of interest into the bacterial cell. Now once you are introducing your gene of interest into the bacterial cell, you will allow them to grow on the master plate or the plate where you are providing everything to them. So basically you will take a plate. This plate will contain agar on it. It will also contain the nutrient rich media on it, and then you will be keeping these colonies here on them. So let's say one bacterial cell here, one here and one here. Now let it grow. Let it form its colony. Let this cell form its colony, and let this cell form its colony. Up till now you are not very sure that which of the given cells have actually taken up the DNA. Now after this, what you will do, once they have sufficiently grown, approximately 9 to 12 colonies or even more than that, when they are grown on the bacterial plate, also you should ensure that they should have quite good amount of distance between them so that intermixing doesn't happen. Once these colonies have grown, now some of the colonies obviously will have the foreign DNA, while the other colonies would not have been able to take up the foreign DNA. Now upon this master plate, you will put a nitrocellulose membrane and you will gently press it on the master plate in a way that some of the imprints of these colonies, means some of the cells from these colonies, come upon the nitrocellulose paper. So we'll take this nitrocellulose membrane. Upon this, some of the colonies will come in this way. Now what you need to do is you need to treat it with detergent or NaOH. Why are you treating it with basic conditions? Because basic conditions will help to remove or lyse the bacterial membrane. Once the bacterial membrane is exposed or removed, the nuclear membrane, although it is not there, so even the cell wall, the plasma membrane, all of them, once they are removed, now only the DNA is left. Now this DNA either can be the recombinant one or it can be a normal DNA, based upon whether the transformation took place or whether it did not happen. Now this DNA you have to fix upon the membrane so that it does not wash away. So now you will either treat this membrane under UV, or you will either bake this membrane at 80 degrees Celsius. Once you do either of them, the DNA will fix permanently upon, or we can say the DNA will stick onto the membrane. After this, you will take the radiolabel probe. Now you know that your gene of interest will have a particular sequence. Suppose it is GATC. Now you will take a radiolabel probe which has a sequence CTAG. Now this radiolabel probe can either be enzyme bound, or this can be a colored probe, or this can be a radioactive probe whatsoever. Now you will take the probe solution and you will introduce the drops on every of the DNA, means whatever here the spots were there, so you will introduce on every of them. Now those DNA which were recombinant and which had that desired sequence, the probe will come and bind on them. and it will permanently stick onto that surface because the actual DNA was also already stuck. So the recombinant DNAs will have the probe bound to them, and the probe will permanently stay upon the nitrocellulose membrane. While the normal DNA, which did not transform, will not have that gene of interest sequence, and the probe, although it will come, it will not have the complementarity and will stay there. Then you will do the washing step. In the washing, what will happen? All the probes will be removed which did not stick, while the stuck probes will remain there to their original DNA. Now suppose after washing you come to know that now you can do anything. If you have the enzyme linked things, then you will give the substrate. Now under the presence of substrate, as we already did this in ELISA, so in the presence of substrate the enzyme will act to produce a colored product. Similarly, you can either treat them with anything like you can do autoradiography, UV exposure, X-ray exposure, whatever, so that the probes will glow. Now only there will be a very small spots only or a fixed spots where the probes are actually glowing, because at all the other places the probes were removed away or they were washed out. So it can clearly indicate that because the probes are present here, definitely means that the original DNA which was present here had the gene of interest, and because of that the probes also stayed here and they were permanently stuck onto this. So now by this you can extrapolate and you can identify yourself that which of the colonies here would be having that complement would be actually transformed, because those colonies which had transformed, only their DNA stuck here, and only their DNA which was stuck here had that probe along with it. So suppose it was colony number 4 and this was colony number 7. So from here we can extrapolate that this colony 4 and this colony 7 were the ones which are transformed colonies, while the other ones failed to take up the gene of interest. So this is how your colony hybridization method works.